of you. Today we are going to study secondary growth in plants. Okay. आज अपन secondary growth in plant या संदर्भात अभ्यास करणार आहे. Okay. So what is actually secondary growth in plant? So before study the secondary growth, let us examine what is actually the primary growth. So primary growth means the growth of root and stem due to apical meristem. That is known as the primary growth. So what is that point? That is, you know that each plant having root system and shoot system. Suppose this is the plant and this plant having shoot. So this is the shoot, and below the ground you can see the root. Okay, and this root having apical meristem, and the shoot, that is the stem, also having the apical meristem. So the length of the plant increasing due to this apical meristem, length of the root increasing due to this apical meristem, root apical meristem. So this increasing the length of the plant is known as the primary growth. So that is the primary growth. Then what is the secondary growth? Secondary growth means suppose this plant after a few years, if examining, the thickness of the plant increased. So this is the same plant, the thickness of the plant increased. Like that, the root also, the thickness increased. So this increasing the thickness of the plant, that means the girth of the plant. Thickness is known as the girth. This increasing the girth of the plant is known as the secondary thickening, secondary growth. So primary growth means increasing the length of the plant due to the apical meristem both in the stem and root. Secondary growth means increasing the girth of the plant, diameter of the plant trunk, shown by most dicotyledonous plant, that is known as the secondary growth. Okay, means dicotyledonous plant is showing this secondary growth, but primary growth is showing by both dicotyledonous and monocotyledonous plant. So see here, this is the young plant. So only diameter this much. So when it grow, it become the diameter increase. This increasing the diameter or the girth of the plant during the growth in dicotyledonous plant, dicotyledonous stem and root, that is known as the secondary growth. So it is very clear, this is the young stem and this is the old one. So the girth is increased. This increasing the girth is known as the secondary growth. So let us study what are the tissue involved in this secondary growth. You know, two lateral meristems are involved in the secondary growth. Means secondary growth साठी दोन टिश्यूचे गरज आहे, that is the vascular cambium and cork cambium. So two lateral meristems. This vascular cambium and cork cambium is known as the lateral meristem. So these two lateral meristem, that is the vascular cambium and cork cambium, is responsible for the secondary growth. So what is actually the vascular cambium? Vascular cambium means, for example, this is the vascular bundle. This is one of the vascular bundle, and this vascular bundle having phloem. So this is the phloem, and below that you can see the xylem. So this is the xylem. In between the phloem and xylem, there is a meristematic tissue, and this meristematic tissue is known as what is vascular cambium. Okay, and cork cambium means the cambium developed in the later stage on the bark, just below the bark. So this is the cork cambium. So this cork cambium and also the vascular cambium takes part in the secondary growth. So vascular cambium, the meristematic tissue that produce vascular tissues, that is the xylem and phloem. So this is the meristematic tissue, and this meristematic tissue produce a phloem outside and a xylem inside. Okay, so the meristematic tissue that produce xylem and phloem are collectively called vascular cambium. So this is the vascular cambium. Okay, in the young stem, it present as a patch in the young stem. Suppose this is the vascular bundle of the young stem, and in this young stem you can see a small patch, a small group of the vascular cambium. Okay, and this cambium later increased and it cover total length of this thickness of this vascular bundle. Okay, so in the young stem it present as a patch, but later it forming a complete ring. So see here, this is the early stage. It is present only in the form of the patch. And when it grows, secondary thickening taken place, it is forming a complete ring. So see here, this is the complete ring. So this is the complete ring of the vascular cambium. So that is the two tissue that involved in the secondary growth. Okay, then let us examine how this ring is formed, how the cambial ring is formed. Let us examine that. So let us look how the cambial ring is formed. That is the formation of the cambial ring. So see, the cells of the medullary race and on either side of the interfascicular cambium, that becomes meristematic and form a cambial ring. Okay, so what is this point? Medullary race. What is actually the medullary race? If you take the TS of the Dicot stem, so suppose this is the TS of the Dicot stem, you can see this is the one of the vascular bundle and this is the another vascular bundle. So like that, number of the vascular bundle is arranged in the stem in the form of the ring. So each vascular bundle having what is intrafascicular cambium. So this is known as the intrafascicular cambium. The cambium present within the vascular bundle, that is known as the intrafascicular cambium. Then what happens in between the two vascular bundle, there are some parenchyma tissue. So this parenchyma tissue is a differentiated simple permanent tissue, and this parenchymatous tissue become meristematic. So here it will become the meristematic, and this meristematic tissue that formed between the vascular bundle, that is known as the interfascicular cambium, and this interfascicular cambium joined with the fascicular cambium or intrafascicular cambium, and finally it here forming a ring. So that is in this way the cambial ring is formed. Okay, so the method, when the stem is growing or the plant is growing, the parenchyma tissue present between the two vascular bundle, that become meristematic, de-differentiation meristematic, and this meristematic tissue joined with the cambium present within the vascular bundle, that is known as the intrafascicular cambium, and the new cambium formed meristematic tissue formed between the vascular bundle, that is known as the interfascicular cambium. So this is the inter means in between, interfascicular cambium, and this is the intrafascicular cambium. Intra means within, intrafascicular cambium. So this intrafascicular cambium and interfascicular cambium that fuse with each other and forming a complete ring. And this complete ring is responsible for the secondary growth. So actually this parenchyma tissue that forming a thin line or the thin thread like structure, that is known as the medullary race. So this is actually medullary race, and this medullary race later become meristematic, and this meristematic tissue become interfascicular cambium, that is the medullary race. So medullary race is nothing but between the vascular bundle there are parenchyma tissue, and this parenchyma tissue is appearing in the form of the thin race, and that is known as a medullary race. This medullary race is a meristematic that combined with the fascicular cambium and forming the ring and forming the cambial ring. This cambial ring showing the differential activity, and that due to this differential activity it is producing the secondary thickening, that is the increasing the girth of the plant. So let us see how the activity of the cambial ring, how the cambial ring is functioning, and what is the differential activity of the cambial ring and producing the secondary thickening. So let us see the activity of the cambial ring, how the cambial ring is acting to produce the secondary thickening, that is increasing the girth of the plant. So see here, this is the cambial ring. So here the cambial ring formation started, and this is the entirely formed cambial ring. So this cambial ring begins to cut off new cells both inward and outward. So this meristematic tissue ring that produce new cells both outside and inside. Okay, नवीन सेल्स बाहेरच्या बाजूला आणि आतल्या बाजूला तयार करणार आहे. So new cells forming both inside and outside. Okay, so the cells cut off towards the periphery, means towards the outer side periphery, that become the secondary phloem. So new cells produced, this cambial ring produce new cells on the outer side or towards the periphery, that become the secondary phloem. You know, always the phloem present outside and the xylem present inside, and that cells produced towards the pith. So this is known as a pith. So new cells formed towards the pith, that become the secondary xylem. So remember, this cambial ring that produce new cells towards the periphery outside, that become the secondary phloem, and towards the pith, that become the secondary xylem. Okay, so the cambium is more active. So if you take the number of the cells produced outside and inside, you can see that the cambial ring is more active towards the pith than towards the periphery. That means more phloem, more secondary phloem present, secondary phloem tissue present outside. So as a result, what happens? The secondary xylem become compact mass. This secondary xylem that is present towards the pith, that become very compact. Means more cells produced, so that applying the more pressure. So this secondary xylem become more compact. Ekdam closely they arrange karna hai. That means it become more compact. And the primary and the secondary phloem. So actually the secondary phloem produced outside. So as a result of this new cells produced, the primary and the secondary phloem that crushed, that become crushed, because the more and more cells producing this cambial ring. So due to the formation of the more and more cells, the second primary and the secondary phloem that is present outside the cambial ring, that become crushed. Crushed means the chura honare, break honare, or it become crushed due to the continuous formation of the secondary xylem. Secondary xylem produces continuously. As a result, the outer cells become crushed or damaged. Okay, the primary xylem remains intact. So when this process is taken place, the primary xylem already present inside, that become intact. The primary xylem remain intact in or around the center. This is the central pith. Around this pith or central part, the second primary xylem that is remaining intact. Okay, so the medullary rays are then formed between the vascular bundles. Medullary rays are formed, and these medullary rays are actually parenchymatous band, parenchyma cells, which pass through the secondary xylem and secondary phloem. See here, this line that is known as the medullary rays. It is passing through the secondary xylem and secondary phloem. Why? Because the primary xylem is remaining intact, and new cells, the new parenchyma rays, parenchyma cells that are producing the meristematic tissue, and that is passing through the secondary xylem and secondary phloem. So that appearing in the form of the rays, that is known as the medullary rays. So then let us see how it further it happens, the thickness increasing. So we already explained the cambial ring producing new cells outside and new cells inside. The cell produced outside, that become the secondary phloem. The cell produced inside, that become the secondary xylem. And this secondary xylem is a more activity towards the pith and less activity towards the periphery. So more and more secondary xylem produce that forcing to form the compact tissue here, and this compact tissue applying a force on the outer phloem, and this phloem, primary and secondary phloem, that become crushed, damaged. Okay, so now this damaged tissue we have to, or the plant have to repair. Okay, so during this repairing, some of the changes have taken place outside, and some of the changes have taken place inside the cambial ring. So the activity of this cambium, you know, this is the cambial ring. The activity of this cambial ring not uniform throughout the year. So this activity is actually depending on the influenced by both the physiological and environmental condition. Means the activity of the cambial ring is depending on the age of the plant, or depending on the condition of the plant, whether the plant is producing the flower or it is not producing the flower. So that is a physiology. So the activity of the cambial ring is depending on two factors, that is the physiological state of the plant, whether the plant is doing more metabolic activity or less metabolic activity, that is a physiological condition. And it is also depending on the environmental condition. So during some month the temperature is very low, and some month the temperature is very high, and some time the temperature in between that, or rainy season. So there are different seasons on the earth, and due to this different season, this activity of the cambial ring also different. So activity of the cambial ring depending on the two factors, that is the physiological state of the plant, metabolic state of the plant, and also depending on the environmental condition. Okay, so due to that, what happens, this cambial ring not functioning uniformly, it differentially performing their activities. Okay, so in the spring, as a result, they are forming two type of the wood plant, that is the spring wood and autumn wood. So let us analyze what is the spring wood and autumn wood. Okay, so in the temperate region means the region of the earth where the temperature is very low temperature, that is the low temperature. In the low temperature region, the climatic conditions are not uniform throughout the year. It is not uniform throughout the year. The temperature will increase or decrease. So in the spring season, during the spring season, what happens, the cambium is very active. Spring season, the cambium is very active and produce a large number of the xylem element having vessels and wider cavities. Vessel producing, angiospermy plant, the most predominant component of the xylem is vessels. Through the vessels, the water is conducting. So during the spring season, this cambial ring is more active and it is producing a more large number of the xylem element, that is the vessels, and that vessels are having more diameter. Okay, the wood formed during this season is called spring wood. So what happened, the spring wood, in the spring wood, cambium is very active and it is producing large number of the xylem vessels with a large cavity, and that wood produced during this spring season is known as the early wood, or it is known as the spring wood. So spring wood is also known as early wood. Okay, so what happens in the winter? In the winter, the cambium is less active. Winter season, the cambium is less active. So as a result, a fewer xylem element producing with narrow vessels. So during the winter season, xylem vessels are very less in number and with very narrow. So this is the xylem vessel during the winter season, and this is the xylem vessel that is producing during the spring season. So spring season diameter is more, and winter season the diameter is less, and the number of the xylem vessel is also less during the winter season, and number of the xylem vessel is more in the spring season. So that is the difference between the spring and winter season, the activity of the cambial ring. Okay, so and the wood that is produced during the winter season with a less number of the xylem and narrow diameter of the narrow lumen of the vessel, and the wood produced during this winter season is known as the autumn wood, or it is known as the late wood. So there are two type of the wood forming. Wood forming in the plant, especially in the temperate plant growing in the temperate region, their condition is, climatic condition is continuously changing. So the wood produced during the spring, that is known as spring wood, or it is known as early wood. The wood produced during the winter season is known as the autumn wood, or it is known as the late wood. Okay. Then let us study one more thing, so that is the heartwood and sap wood. So before we deal with heartwood and sapwood, one more point, that is the annual ring. So what is actually the annual ring? Annual ring means, especially in the temperate region, plant growing in the temperate region, it produces two type of the ring. That is the wood produced during the spring season and wood produced during the winter season. So in the spring season, it produces the wood that is actually known as the early wood, and this early wood is lighter in color. So color is very light. So suppose this is the early wood. It is a light in color, not a very dense. So suppose this is the tiers of the stem, and you can see the spring wood. So this is the spring wood. The spring wood or the early wood, it is very light in color. So it produces during the spring, and when the autumn or the next season, that is the autumn wood, it produces in the autumn wood. This autumn wood is very dark in color. So spring wood, early wood, and autumn wood, it is a very dark in color, because the deposition of the some of the chemical, not only chemicals, but also it is due to the more cells produced. So it become very dense. So spring wood is a lighter, light colored. Spring wood is a light colored and it has a lower density. Okay. Autumn wood having darker and has a higher density. So here two ring formed. So this is the spring ring and this is the autumn ring. So in a year, a plant having two rings, that is the spring wood and autumn wood. This autumn wood and spring wood, you can see two ring present on that. These two ring together constitute 1 year of the plant. So 1 year of the plant. That means in each and every year the plant produce two ring, that is the spring wood and autumn wood, and these two ring is known as the annual ring. So this annual ring, when you count the annual ring, you can count the age of the plant. So if 10 annual ring present, the plant having 5 years, because in each and every year it is producing two ring, that is the autumn ring and autumn ring and spring ring, okay, or autumn wood and spring wood. So it is producing two ring and two ring together forming 1 year. So counting the number of the ring, you can count the age of the plant. So that is known as the annual ring. So here, see here, this is the ring, and this ring count and how many ring present, just half that is the age of the plant. Okay, so that is the annual ring. So let us go the next point. So let us see another two type of the wood, that is known as the heartwood and sapwood. So what is actually the heartwood and sapwood? See here, in older trees, when the tree is very old, greater part of the secondary xylem is dark. Okay, so where is actually secondary xylem present? Suppose this is the TS of the plant, and this is the cambial ring, and this cambial ring produces secondary xylem in the towards the center and a secondary phloem towards the outside. So this is the phloem, secondary phloem, and this is the secondary xylem. Okay, so see, secondary xylem is producing inside. So in the older trees, what happens? The greater part of the secondary xylem is a dark brown due to the deposition of the organic compound. Some of the organic compound depositing on the secondary xylem. Okay, so what are these organic compound? This organic compound may be tannin. Sorry, it may be tannin, or it may be resin. Tannin or resin, or both resins, gum, or different type of oils, or gum, sorry, gum, or aromatic substances. Some of the aromatic substances. This substance depositing in the secondary xylem, aromatic substance. That different type of the aromatic substance. We cannot take one large number of the aromatic substances that is depositing on the secondary xylem. So what happens? This secondary xylem become very dark in color. Okay, this substance make the wood present inside, that is inside the cambial ring on the secondary xylem, that it become very hard, and also it become very durable, so long lasting and resistant to attack microorganisms and insect. So see here, the inner part, that is the secondary xylem, and in that secondary xylem all the chemicals are deposited, and it become very dark, very hard and very durable, and that cannot attack by the insect or the valvi, mungi, attack bacteria, nothing, that it cannot attack on this. So that is very durable, and this part, the hard part, so later change into the part of the plant that is known as the heartwood. Okay, so that is known as the heartwood, and this heartwood, due to this deposition of the different chemicals, it cannot conduct water. So heartwood does not conduct water, but it gives a mechanical support to the stem. It gives mechanical support. So plant la mechanical support, it cannot conduct water. Why it cannot conduct water? Because it is already dead, and because all the vessels and the tubes are deposited with these aromatic substances, tannin, resin, oil, etc. Okay, so the peripheral region of the secondary xylem. So this is the primary xylem part, secondary xylem part. The peripheral region of the secondary xylem is lighter. So this part, it is very lighter. See here, this part is very lighter in color and is known as the sapwood. So this is known as the sapwood. So this is the sapwood. So the sapwood and heartwood is actually depending on the, or it is related with what is xylem. So secondary xylem is a light. The peripheral region region of the secondary xylem is a lighter color and known as the sapwood. So it containing vessels and it is living and it can conduct water. So it involved in the conduction of the water and minerals from the root to leaf. So two type of the wood forming. That is inside the cambial ring, that is known as the heartwood in the center that cannot conduct water. Outside the heartwood you can see the lighter area that conduct water and that is known as the sapwood. So heartwood and sapwood both are related with the xylem. Okay, so that is the heartwood and sapwood. So let us examine how the secondary thickening, that is the increasing the girth of the plant. So up to that we cannot mention how the thickness is increasing. Only the cambial ring formed, secondary phloem formed outside, secondary xylem formed inside. This secondary xylem or the tissue present inside the cambial ring that forming two type of the wood, that is the heartwood and sapwood. So all these we mentioned, winter early wood, late wood, all this we explained. Then how exactly the thickness of the plant is increasing. Okay, so let us see the Cork Cambium. So actually it is responsible by the Cork Cambium. So let us say what is actually the Cork Cambium. So see, as the secondary thickening is continuously taken place, so the secondary thickening continuously taken place, the girth will increase. As the secondary thickening continued, the outer cortical and epidermal layers become broken. See here. So secondary thickening is continuously taken place. New cells forming outside secondary phloem, new cells forming inside secondary xylem. So due to this continuous formation of the new tissues, a pressure is applying on the outer side. Okay, epidermal layers, a pressure applying on the epidermal layers. This pressure, due to this pressure, the broken layers needed to replace. The tissues will break due to the pressure, and these broken layers must be repaired. So during the repairing, some of the changes have taken place outside the or within the epidermal layers, within the epidermal region. Some changes have taken place. So what changes have taken place? Let us see. So what is the requirement here? The outer tissues are broken, and this broken tissue must be repaired. बाहरचे सेल्स डॅमेज आले सेल्स आपण रिपेअर करायला पाहिजे. रिपेअर करण्यासाठी तिथं काय तयार झालं पाहिजे? तिथं एक पर्टिक्युलर मेरिस्टेमॅटिक टिश्यू तिथं तयार व्हायला पाहिजे. सो ए टाइप ऑफ द मेरिस्टेमॅटिक टिश्यू फॉर्मिंग आउटसाइड. For what purpose? The cells are broken, the tissues are broken. To repair that broken tissue, a meristematic tissue layer is forming outside. That meristematic tissue formed is known as the phloem. Okay, so a cambium, Cork Cambium is forming, and that meristematic tissue is known as the Cork Cambium, or it is known as the phloem developing. Suppose this is the TS of the plant. Okay, so the changes we already mentioned here, that is the heartwood and the sapwood. So new tissues continuously forming outside and inside also. As a result, this outer part that become crushed. So the outer part crushed, and this damage should be repaired. So for the repairing of this damage, a new layer of the cambium is forming here, meristematic tissue forming, and this new meristematic tissue formed, that is known as the phloem. And this phloem is also known as the Cork Cambium. Okay, it is also known as the Cork Cambium. And this Cork Cambium, you know, it is a cambium, or it is a meristematic tissue, and this meristematic tissue again producing new cells outside and new cells inside. So new cells forming outside and new cells forming inside. That is, it is a differentially producing cells both outside and inside. The outer cells, the new cells forming outer side of this phloem, that is differentiated into cork, or it is producing the phloem. So outer side, what is forming? Outer side cork is forming, cork, or it is known as the phloem is forming, phloem. It is also known as the phloem. So remember, due to continuous production of the new cells, the outer cells become damaged, and this damage should be repaired. For the repairing, a new cambial ring forming. That cambial ring formed is known as the phloem, or it is known as the Cork Cambium. this cork cambium produces new cells outside that become phloem, or it is known as the cork, and new cells produce inside. Some of the cells produce inside also, that is differentiated into phyloderm. So inside it is producing the phyloderm, or it is known as the secondary cortex, phyloderm or secondary cortex. So remember, this is the phylogen producing outside cork or phloem, and inside phyloderm or the secondary cortex. So here this phylogen, so the phylogen is the meristematic tissue that is producing phloem outside and phyloderm inside, and all these three layers together known as the periderm. So it is known as the periderm. So like that, periderm is formed, and during this formation of the phylogen, phloem and phyloderm, the thickness of the plant automatically increase, and that activity in the spring is known as the early wood, and that activity in the winter is known as the late wood. So two ring forming here, that is the light ring and dark ring, or the soft ring, or it is producing the two ring, and this ring early wood and late wood, that is forming two separate ring, and that ring representing the 1 year of the growth. So counting this, that is annual ring, counting the number of the annual ring, you can count the age of the plant. Okay, so that is the cork cambium. So we already explained phylogen, phyloderm and phloem. Okay, so phylogen present in the middle, and outside phloem, this is the phloem, and inside what is present? Inside the phyloderm, and all these together known as the periderm. Okay, so due to the continuous activity of this phylogen, so see here, this is the phylogen. This phylogen is actually meristematic tissue. So continuous activity of the phylogen, new tissues are continuously producing, and that exert pressure and develops on the outer layer of the stem. It is producing the pressure on the outer layer. As a result, what happens? The outer layer that become die and cut off, and bark is formed. As a result, the bark is formed. Okay, the bark refers to all tissues exterior to the vascular cambium. So suppose here is the vascular cambium, here is the vascular cambium. So this is the vascular bundle, this is the vascular cambium. So all tissue present outside this vascular cambium from the epidermis up to this point. So that include what is secondary phloem coming, primary phloem, that including, so that including, because all the tissue present above the vascular cambium. So that including the periderm. Periderm is phylogen, phyloderm and phloem, and also that including secondary phloem. Remember, the bark means all the tissue present outside the vascular cambium. So vascular cambium, above the vascular cambium is you can see the phloem and secondary phloem, then phyloderm, then phylogen and phloem. So all the tissues, periderm and secondary phloem together constituting what is, that is the bark. Okay, these bark are two types. That is the bark that is formed in the early season, that is the spring season, that is known as the early or the soft bark, and the bark produced during the winter season, that is a later stage, that is known as the hard bark or late bark or hard bark. So actually the bark is bark during the formation of this bark, it crushed, it produced some lens-shaped region that is known as the lenticel. Let us see the lenticel. So see what is actually the lenticel. Lenticel means when the secondary growth taken place, new tissues are continuously producing outside, and that exerting the pressure on the bark, and as a result the bark will break. Okay, so the epidermis is broken, and complementary new cells are formed here. So this is the new cell formed. Why new cell formed? To repair the broken epidermis. So this new cell formed, that is known as the complementary cells, and just below that you can see the cambium, and this cambium is known as the phylogen, or it is known as the cork cambium, and below that you can see secondary tissues, secondary cortex. This is the secondary cortex. So this is secondary cortex later formed, and this is the phylogen or the cork cambium, and above the cork cambium it produces some cells to repair the broken part, that is the complementary cells, and here the bark is broken. Okay, the broken bark, it is showing some of the lens shaped structure. See here, so these are the some of the lens shaped structure, and this lens shaped structure is known as what is, that is known as the lenticel. So that is the lenticel. So what is the function of the lenticel? These lenticels helping to exchange oxygen and carbon dioxide. So gaseous exchange is the function of this lens shaped structure. It is helping to exchange gases. It helping or it permit exchange of the gases. So that is a function of the lenticel. So like that lenticel is formed. So that is actually the secondary growth in the stem. Let us see the diagram that is showing the secondary growth. So this is the diagrammatic explanation of the secondary thickening or secondary growth in the case of the stem. So this is the first stage here, vascular bundle. And this vascular bundle having the cambium that is known as the fascicular cambium or vascular cambium. And the tissue present between the vascular bundle, that is the parenchyma tissue, and this parenchyma tissue differentiated into patch of the meristematic tissue, and that patch of the meristematic tissue become the cambium between the vascular bundle, that is known as the interfascicular cambium. And this interfascicular cambium and the fascicular cambium that is joined with each other and forming a ring of the cambium. So this is the cambial ring, and this cambial ring is differentially active depending on the environmental condition and the physiology of the plant. Okay, so during the formation of this interfascicular cambium, the parenchyma tissue present between the vascular bundle that is forming a ray-like structure that is known as the medullary race. So see here, this is the one medullary race, this is another medullary race, and this medullary race passing through the secondary phloem, secondary xylem, and even primary xylem. But this is a short one shown that passing secondary xylem, secondary phloem, and a primary phloem, but it is not reaching to the primary xylem. That is the reason why it is shown one less length and another one is long, small and short and long. Short one reaching up to the secondary xylem, and the long one reaching up to the second primary xylem and secondary xylem, even secondary xylem. So that is shown here. And here when it is happening, here forming new tissues continuously producing both inside and outside. As a result, the inner part become very compact and that is depositing tannins, gum, and some organic materials, and finally it become very dark in color and very strong, cannot attack by the insect or any other infections, and that hard part that is known as a hard wood, and outside you can see that containing the primary xylem and secondary xylem present. Outside that is very soft, that is soft wood that conduct water, hard wood cannot conduct water. Simultaneously some changes taken place outside the cambial ring, that is a new tissue forming that is differentiated into secondary phloem, and this secondary phloem continuously increasing that is causing the breaking of the epidermis, and this broken epidermis must be cured or must be repaired for that new cambium forming that is known as the Cork Cambium. Cork Cambium producing new cells, that is the complementary cells that also producing new cells inside, that is known as secondary cortex. And finally it here forming a small opening, a lens shaped opening, that is known as the lenticel. So that is the secondary thickening in the stem. So let us analyze what happens during the secondary thickening in root. So let us analyze how the secondary growth taken place in the case of the Dicot root. Okay, so what happens in the Dicot root? The Dicot root. So what happens in the Dicot root? So see here, this is the TS of the Dicot root. Okay, so see here, this is the xylem. It is a tetrarch. This is another xylem. This is the another xylem, and this is the fourth xylem. So you know the phloem is present between that. That is a radial type arrangement of the vascular bundle. We already explained anatomy of the Dicot root. Okay, so what happens? The cells present here is the pericycle. Suppose this is the pericycle. The cells present just below the pericycle that become meristematic, and this meristematic cell that newly produced, it forming a wavy ring around the phloem. Okay, and then it moving down below the phloem, and then above the xylem, below the phloem, above the xylem, below the phloem. So like that it producing a ring. It is producing a ring here. Okay, and this ring is actually newly formed cambial ring, and this cambial ring is totally secondary in origin. So Actually the cambium is absent in the case of the root, so it is a closed type vascular bundle. But during the secondary thickening, the cells present just below the pericycle that become meristematic, and that producing new layer, and this new layer is passing like this. That is the below the phloem and above the xylem, below the phloem, above the xylem, and forming a wavy ring. And this wavy ring formed, that is actually known as the cambium, and this cambium produces new cells outside and new cells inside. So automatically this phloem that is moving above or outward, and it applying the pressure on inside, and inside become a pith, thick, dark, and very dense region that becomes the pith. And from this point onward, all the changes that taken place is similar to the dicot stem. So only one difference between the dicot root and dicot stem secondary growth. In the case of the dicot root, a cambial ring is formed, and this cambial ring is actually forming from the cells present just below the pericycle, and that forming a wavy cambial ring, and this wavy cambial ring having the xylem inside and the phloem present outside. And so then it is producing new layers that is very much similar to the dicot stem, that is the phalogen, phalloderm, and cork, lenticels, all the changes and that have taken place inside, pith forming, very dense region forming, very resistant region forming, all these are the very much similar. So here the very important point, all the secondary, that is the vascular cambium formed, is totally secondary in origin, because vascular cambium was absent, totally absent. So here the vascular cambium formed between the phloem and the xylem, and it formed a later stage, that is during the secondary growth. So this vascular cambium is purely secondary in origin, because it is forming only during the secondary growth. Otherwise there was no cambium present between the xylem and phloem. So that is the secondary growth. So this diagram very clearly explaining the same point. That is the tetrarch xylem. In the case of the root, the xylem is a tetrarch. So these are the xylem, and between the xylem you can see the phloem, and a new layer forming, new cambium is forming around that, and that producing new cells both outside and inside. So that is the secondary growth in secondary growth in root, that is a dicot root. So let us conclude that secondary growth in dicot plant, both root and stem showing the secondary growth, but the secondary growth in the case of the stem is more predominant comparing to the secondary growth in the root. And only difference is that in the case of the root, or the cambium ring formed is totally secondary in nature, but here it is already primary cambium present between the xylem and the phloem. Because the vascular bundle is open vascular bundle, in the root the vascular bundle is closed vascular bundle. So that is all about the secondary growth in dicot root and dicot stem. So this is the dicot root, and this is the dicot stem, secondary growth in the dicot stem. So that is